And that's the theme of my talk, really, is to sharpen the precision with which we can characterize proteins that are created from the 20,300 or so human genes. And so by domestication, what I mean to say with that metaphor is, it's not that you understand everything when you domesticate something, but you sure can interact with it deterministically and regularize it and use it, exploit it for the improving the human condition. So my goals with you and for you are to offer a perspective about the human proteome, mainly the composition of it, which isn't mapped with high precision, and we can do better, and I'll try and make the case for that with you today. So the compositional complexity, just what a chemist would think is a molecule, can we get very precise about that? And then I'll assert that we can regularize protein-level biology better through better measurements, and that the functional assignment of post-translational modifications is actually inefficient, and that we can ratchet that up. And I know, like Megan and others are intensely interested in this. So I offer you my new hobby as a springboard. And Richard, Scialog has had many astrophysics and precision cosmology, physical astronomy. It's been an incredible 25-year run in this field. And I have internalized the lessons from this field. I just watched every YouTube video almost, so I'm an expert, on... And I give you the Hubble ultra-deep field and, just an amazing set of lessons learned here. And so let's go through them, the things that I've learned recently, that if you have this complex system and, let's just measure it precisely. And eventually you will get clarity and an epiphany. So I start way back with a Danish nobleman, Tycho Brahe. He had an instrument that looks like this. This was his data from Mars over about 15 years. And, not more than 100 years later, sun at the center of the solar system. Amazing effect of what he was doing. More recently, you have the Edwin Hubble and the Mount Wilson Observatory, not far from here. You all know the story. And, this didn't require log-log plots or semi-log. This was linear. The distance from a Cepheid variable star and the rate at which it's receding away from us. And so this was the Hubble's law, and Andromeda was seen as what it was, not a nebula, but a new galaxy. And so the universe expanded by 3 orders of magnitude, right? Not a million light years, but a billion, 10 billion light years. Much more recently, the Keck Observatory. This one you can't even really see. There's 2 lines here, but that's the difference between a big crunch and a big expansion and a fizzle to the end of our universe trillions of years from now. And that actually the expansion of the universe is accelerating. And using type 1a supernovas as a standard candle led people to match this upper line and then hit a Nobel Prize. And so theme, better measurements, with great instrumentation, yields data, and ultimately epiphanies. So let's turn now inward to us and think about mapping the protein universe within us. And the machinery of the cell, when I hear the chemical machinery, I think protein nanomachines are a critical element of that. And arguably one of the top things you'd like to wrestle down, domesticate, regularize. So let me just sort of, in pretty quick style, update you on the field, the wild and wacky world of proteomics. So here was a 9-figure investment from the Knut Wallenberg Foundation based in Stockholm. It's called the Human Protein Atlas, led by Matthias Uhlin and Emma Lundberg. And we've got now 12,000 antibodies validated, ever-increasing quality with the antibodies. This has been about a decade-long run. And, you're mapping cytoskeleton, nucleolus, subcellular localization of 12,000 proteins using antibodies. And so it's this beautiful atlas. And it's separated in space. And so that's normally what people think of as an atlas. I'll define a new type of atlas in a moment. But this shows what big science can do and is moving. And I'll come back to the HPA in a moment. Also in the news, the past 2 years has really seen a sea change in mapping human cell types. And this has been elevated out of the Common Fund and now in San Francisco with the Chan-Zuckerberg group and run out of the Biohub. The Human Cell Atlas now definitively answers the question, which is a provocative one. Why do this, right? They have a 71-page PDF article that just lays out with vision and leadership how to regularize an open frontier, like cell types. And you could say, well, I have 1 trillion cells in my body. I'm 5 feet tall. Maybe I have 500 billion cells in my body. Everyone's a snowflake, right? Well, but we're scientists, and we should be able to bin and regularize. We did it with life on the planet, right? We regularize complex systems. So I'll come back to the HCA as well. Here's already bearing fruit here. This is in press at Nature. Steve Quake's leading this, fueled by the billions put in by Mark and his wife. And so here's, if you're not into t-SNE plots, here's a pedagogical moment. I'm getting into these t-SNE plots. This is like a fancy PCA, but more clustering, like to have local separation in data. And so here's then all the organs of a single mouse. They all teams took it and did single cell RNA sequencing. That's the underpinning technology for the Human Cell Atlas. And so here's 40,000 cells, the results, and put into high dimensional space, thousands of transcripts for each one of those single cells, and then mapped into this t-SNE plot, T-S-N-E plot. So it's moving, and this is a template for what will be done in human. And you can say, well, there's a lot of cell relatedness. This is really quite clean, microglia. Maybe there's some structure within here that might reveal biology. Here, much more overlapping in mesenchymal cells, et cetera. So we're going to regularize that world, and now you can bank on it. And I think that's a provocative idea. It doesn't get support to automatically do it. In fact, Sidney Brenner in the 90s was calling for the Human Cell Atlas. But the time in history, the arc of history really... question? Yeah?
Why are B cells and T cells so far away?
I will defer to Steve Quake. You can ask this gentleman. Yeah, so it's a high-dimensional space, and there's a beautiful YouTube video on t-SNE plots. A Dutch fellow invented it in 2008, and it really does separate out high dimensionality and. Yeah, it's complicated. You're right. Okay. So I'm imagining, and I know Emma Lundberg, who's taken over the HPA, Human Protein Atlas. She's going to put, and she's in residence at the Biohub, and I believe we're going to put these two things in register with one another. So you'll have cell type information, and then within the context of however many thousands of cells in this ontology of human cell types, you will be able to look at some proteomic information, and we'll merge these two. And that's fine. That's separating in space mostly, which is the natural way that people map, right? The map of a city is spatially resolved. Map the body. Got it. Okay. With cellular and molecular resolution. So I'm going to make the tougher case of what a compositional proteomics experiment might look like, or a project. And so what would that look like? And so let me back up to the genome, starting to accelerate a bit. That was about the composition, 3 billion letters, $1 each. That's how much it cost by 95, 96 to sequence. That was $3 billion. And the compositional, that's the exception that proves the rule. That's where compositional mapping was so critical. And, but then it led to NGS and all the other types of sequencing technologies. And you can now map like the 4D nucleome in space, or a cancer patient through time. But I'll just remind us that we did not do 1,000 cancer genomes project before we did the first 27 people correctly. Like we wrestled the measurement problem to the ground, and that was expensive. But then through a very intentional approach by the then launched NHGRI, National Human Genome Research Institute, you had a decade, and I'll show you a drop in price graph later, but that's sort of the lesson of the history, right? So let me use that across the central dogma and bring us past a lot of other big science efforts, medium to big science efforts, skipping through genomics and focus on proteomics, and that the HPA is a tissue-level map, and they're going subcellular now. 2 science papers describe that effort. But I want to focus on this compositional part, because for whatever stroke of fate or luck or I don't know, that's what I'm good at. And I'll just give you a state of play in that sphere, this mapping protein composition, full atom composition. Okay, so I'm going to give you a tri-level framework in which you can think about things. There they are. So the bottom-up proteomics, this is the majority dominant technology, usually by trypsin or other proteases. You digest a big complex mixture, like a proteome, whole proteome or bodily fluids, and you create millions of peptides. And I like the nursery rhyme Humpty Dumpty from English folklore. It's hard to put it all back together again. And I'll have I have a lot of great friends who work in this field, and it's the dominant technology. It's done some amazing things. I'll review them in one slide, and we'll move on. But there is an alternative now, and that's to not use trypsin. And it's to go directly at what we'll come to call proteoforms. And you can even attack at this top level. You can take whole protein complexes, whole nanomachines, fly them in the gas phase, and get complete atom composition of what's there. Okay, and so this is starting to take some hold, that actually, on a per measurement basis, the value of the measurements, it can increase as you go up. Like the underlying biology is really revealed with more faithfulness. Bless you. And that's an emergent idea. And I also have been advancing the idea at Northwestern, the use of proteoforms as biomarkers, and to revive interest and excitement around statistical correlations to complex phenotypes. Because I believe that the measurement approach that the world took in 2000 and beyond degraded the statistical power of the measurements, and that can be revisited. All right, so it's a simple idea, and metabolomics is done top-down, right? You wouldn't say, hey, I've got a mixture of 2,000 small molecules in the human body, and I'm going to run this profile. But wait, before I do that, I'm going to degrade the covalent bonds between the atoms and create this puzzle, and then, don't get nervous, then I'm going to lose 80% of the atoms, and then we're going to infer what happened. That's the world's best technology, well, the widespread technology, yet we're facing this kind of complexity. So we all know there's a canonical base sequence. That's what UniProt, the UniProt consortium calls it, the base sequence, upon which we decorate. And this, we'll spend a lot of time talking about PTMs, but there's other sources of variation that occur in combination. That's proteolysis, alternative splicing, which creates isoforms, different base sequences. And then the combination of mutations with all of this variation, can we get more precise about this? Because it should be fairly obvious to most of you that can be hard if you digest this, which we'll do in 2 slides, to put it all back together with precision. So bottom-up proteomics, as I mentioned, amazing, right? I mean, it's a $3 to $4 billion a year marketplace, depending on how you monetize. You can now do APMS or IPMS, affinity pull-downs, map complexes at scale. Steve Gigi and others, Steve had a great Cell paper recently. And we're starting to get great network diagrams about the interconnectivity within cells. Peptide correlation profiling. So this is like Catherine Lilly or Rudy Aebersold. This is PCA, and it's just cofractionation is the concept. So if I wash a lysate across a column of some type, I look for co-fractionation of peptides, and I infer either organelles or complexes from doing that. And then the field's all lathered up to go after single-cell proteomics. Fun, a lot because the stakeholders, whether they be federal or foundation, are very interested in single-cell genomics, and that's sort of pulling proteomics into that, despite the fact we don't have PCR. But there's a lot of people really pushing below 1,000 cells and in the next 5 years trying to get to be able to do that at some depth of the proteome. And then disruption happens all the time in fields. So Ed Marcotte, just 2 weeks ago at a conference, revealed his publication, which will be out like next week probably in Nature Biotechnology, where he uses basically single molecule Edmund sequencing on a chip. And he can start to get partial sequences, and with a DNA inferred protein landscape, actually start to identify up to 30 or 40 cycles of Edmund sequencing on a chip. So it's a great time to be alive. So so my Humpty Dumpty analogy, right? The inference problem. People in my field don't like to talk much about the inference problem. I love to talk about the inference problem. It's because there is a solution to it, where you can have gene-specific identifications, isoform-specific and proteoform-specific identifications. And so I'll cartoon it for you. If you do a trypsin digestion of 2 proteoforms here, you can get artificial loss of PTMs upon digestion. And then things go right. There's actually some dark matter in this universe, as I kind of hinted at. You can lose up to 80% of the peptides. And if you don't do deep proteomics, which is to say expensive proteomics, you exacerbate the amount of dark matter in the universe in which you're living. And then you have to try and stitch Humpty Dumpty back together again. And it's basically impossible to definitively reassemble the peptides and come out with a decent inference of what was there. And that reality is, I think, reaching a discussion point and an acceptance within mass spec-based proteomics. So it's good to be making good progress toward your goals. And don't give up. This has been 20 years for me, and I got 20 more to go. You can directly read out the intact mass, and I have in the Q&A some slides that can take you through this, or tonight in the bar, and Bill's buying the gin and tonics tonight. So this issue, like PTM combinations retained, or how specific alleles, allele-specific modification patterns, which are on specific isoforms. That's a really subtle, context-dependent question, and we can access it now, and I'll show you an example of that on KRAS. Just to finish off the context here, and there in 2014, 2 papers dropped that had a first draft of the human proteome at the biofluid and tissue level. 2 groups, Akilish Pandey and Bernard Kuster, and their groups did this. But as you now know, peptides aren't the proteins themselves. They only capture a portion of the variability, and they don't correlate really well to phenotype. So as a unit of currency for the field, I think we can do better, and we can close knowledge gaps in the process. Okay, so then and speaking with Dan just before the talk, he's like, well, Neil, tell people a little bit about the headwinds that you faced in putting out the scope of being able to get the composition of the proteome reasonably wrestled down. And I would say, yeah, the headwinds, I got an assistant professor job at the University of Illinois, and the theme was weigh every protein. I went, weigh every protein. Got me a job, and good to have a tagline. And in 2012, we got resources from vendors that were active in the space, or from foundations, or NIH provides conference support to get together. And we coined a term at that meeting in 2012 in Lisbon, Portugal. Took about $40,000, and we formed a consortium, a research consortium, which is now a 501c3 based in Boston, the Consortium for Top-Down Proteomics. We got a tagline, bringing proteoforms to life. And we've codified, okay, here's a glycosylation with a phosphorylation and a SNP, and that exon is in the protein. So that combination of things is now called a proteoform, and we're putting them in databases. We have PFR numbers, proteoform record numbers, And we're doing it as a community. So now with all of that as preamble, you can now look at some actual data. And so here's a gene, and we have two isoforms, long and short, from alternative splicing. And then we're going to spend a lot of the rest of the time on the proteoform explosion and what people's perceptions of that are. Like, how do PTMs, of course, they exponentially increase the complexity of the proteome, but by how much? So that's what we'll get to. For now, just take some lessons away from this low bias readout at the proteoform level. You separate isoforms, and here's a phospho and acetyls, and you can get the hierarchy, like maybe a phospho is needed for an acetyltransferase to write an acetyl mark on the protein, and that's very doable with top-down, up to about 50 kilodaltons in denatured mode. So what I just showed you was a spectrum from electrospray mass spectrometry, and here's a 47 kilodalton protein, and you get all these charge states. These are all just different protonation states, lysines, arginines, and histidines at acidic pH, and that's the the world of intact mass, whole protein mass spectrometry using LCMS, or electrospray mass spectrometry. So we'll focus on this mode, and then we'll switch to native mode. And there's a lot of excitement about native mode, and you can go to much higher mass, and that's where you can really attack, the molecular machines and get their composition. So through the consortium, like one of the active board members in the consortium is Ying Ge at University of Wisconsin, and she's been for years talking to the cardiovascular community, CVD research, and she's got just so clearly a bird's eye view of what happens in the progressions of heart disease. This is true of cardiac troponin in human and pig hearts, that as you get towards congestive heart failure, you get a massive decrease in these phosphoproteoforms. And there is actually a hierarchy. One of the phosphos has to fire first before the other, by and large. And she's worked all this out. So I'm toying with this metaphor. We'll try it out on you. Give me votes about what you think about this. But top-down proteomics is a filter. So in comes complexity, and out comes what people want, which is understanding and interrelationships of these sources of variation. And there's reasons why I believe it's worth pushing the effort and doing this. And one is, like we saw last night, the stoichiometry. So one of the reasons I'm positing this as a filter for function, value, and efficiency to assign function to these PTMs and measure them with more rigor is that, you can basically see things that are above 1% stoichiometry. It's usually a threshold, 5% if the signal's not so great. So that is a different view than what you get using peptide-centric approaches. And I've got some ancillary slides if you want to drill into that, but they really measured fundamentally different things. Peptides, whole proteoforms. And so you have a different view of the biology through this filter. And like any filtration process, you got to really want it and stuff it through. I will say that the community has changed drastically in the last 4 to 5 years. The private sector has moved the ball tremendously. There are commercial solutions for this. Certainly wasn't the case even 4 or 5 years ago. So I'm going to, how are we doing on time there, Richard?
You're good.
Yeah?
Yeah, you got easily another 5 minutes.
I'm just worried when if there's a snake coming or maybe the cane. Yeah, so the RAS, so if you're into technology development, it took me 20 years to really figure this out. I'm a slow learner. Nothing was more impressive to me than the effect of picking a great problem, burying your head in it, and now, RAS biology, there's a whole RAS initiative in the NCI run by Frank McCormick, and forget about all the general speak that I gave you. Just show it in a system where people are really hyper-focused on it. Probably obvious, you've probably figured all that out. I'm a little slow. But in RAS biology, there's 3 human genes. There's 2 isoforms from KRAS. creating two very different C-terminali in the hypervariable region. This is like 95 to higher % identity. So when you digest this, you get all sorts of imprecisions, and you basically shred the C-terminus. So now you can't access, the biology of any given isoform, and there is isoform-specific biology in KRAS 4B versus 4A, absolutely. And so working with a national infrastructure is another way to advance your cause. In this case, the National High Magnetic Field Laboratory in Tallahassee, Florida, they have a monster $12 million mass spectrometer, and it just reveals the proteoform landscape quite readily. And so we've mapped about 25 proteoforms of KRAS 4B and 4A, and published a paper on it. There's a hypothesis that KRAS 4A is exceptionally toxic and driving proliferation. KRAS is mutated at 3 different sites with up to 20% of human cancers, pancreatic most notably. So if you want just a one-page summary of what I told you today for the inference problem and the whole deal, this is a good reference, just a one-pager, one figure. I just want to focus for today on the protein network, and they're usually shown as single dots, right? But now they explode into a proteoform family that needs to be mapped and can drastically regulate the functions of proteins in that network. And it very much affects cell fate and decision making. And so, mapping KRAS really got the RAS initiative and NCI more knowledgeable about proteoforms. So let's now spend the remaining time trying to answer this question. Because imagine a bag of cats. There's about 20 of them in the bag, and then you let them go. This question does that, even to practitioners of proteomics and consumers of proteomics information. This question spreads people widely. So there's a 5-page paper. I got together 60 close personal friends and said, let's ask this question. What do you think? And 4 months of your life later, you're trying to integrate all these ideas, and we've tried to answer the question, but the perception of many is as follows. There's a lot of variability. PTMs make things explode in complexity. But it's been our experience from looking down from the top, bird's eye view on proteins, that biology is constrictive. That gifted to us through evolution is some really good programming. So think about the histone code hypothesis, think about other experiences about how PTMs are laid down, how they're written, erased, and read. And so what are the models for the proteome complexity? I've graphed it just in my brain, so here's 20,000 human genes and different models of what the explosion could be. And I've got 2 examples where you look at enolase and histone H4. And there are hubs of cellular integration that have lots of modifications, P53 and F-kappa B. Those are tough. Those are going to be, up in the extreme world. But there's a lot. Let me just show you 2 examples, okay? And my position will be clear. Histone H4, we know the answers, basically. And if you go into human Swiss Prot, in the UniProt consortium, there's the accession for histone H4, 66 possible PTMs, 17 sites, cosmologically large number of potential proteoforms. How many are actually made? And in what cellular contexts? The answer, it will appear on the next slide. And I just give you a smaller subset, because there's a lot known, right? So let's hit the acetyls, let's hit the monoditrimethyl on lysines here, and some other phosphos and the one SNP that's known. You do the math, 98,000 possible proteoforms. The answer is actually 75. But within what detection limit? Okay, so 7 different labs in the consortium. We sent around H4. This is integrated over about 7 different cell lines through the cell cycle, deal with the DNA damage happening. That scope of biology, all cancer cell lines, down to over 4 logs of dynamic range. Okay, so you have about 50 to 80 million nucleosomes in a diploid cell. Half of them in cancer cells are this proteoform with N-terminal acetyl and lysine 20 dimethyl. And then you go down 4 logs in dynamic range, about 4.5 logs, and there's only 75. So that's what there's a prescriptive nature of the writing and maintenance of those marks. Now, we can have a great conversation, and I sure hope we can at Scialog, about this part of the iceberg. You know, I'm sure there's really important biology down there, right? Like just at a few transcriptional start sites of an active or repressed promoter, very rare marks. But also down there is, histone acetyltransferase is having a bad day. Like everyone's got a bad day, right? So stochastic biology. Maybe you grab propionyl-CoA instead of acetyl-CoA, and you put a propionyl group on a lysine, right? Everybody has a bad day. So but I guess the major takeaway from today is that, map things that are present, and that, that's the takeaway. So let's just go to enolase and go to native, and then I'll move to close. So let's go top down, or call it whatever you want. It's a really high value mode of electrospray, and it reads out the machines that are just present endogenously. You still need millions of cells to do this in an untargeted way. Most of the people do it in a targeted fashion, like structural mass spectrometry, complementary to X-ray and cryo-EM. That's mostly how things go, and so I resonated very much with your talk last night about going a bit more untargeted, and you actually get much fewer charge states for native mode, and that allows signals for any one of them to be higher. And you can go up to whole viruses. You can go up to 10 megadalton, even slightly higher with today's technology. So and there's this beautiful thing that happens in the physics of this. So you spray it, sort of an electro sneeze into the gas phase. The fidelity of the readout of information is very high. So metal binding, cofactors, stoichiometry of complexes, it all is retained with high fidelity. So here's a tetramer. You then tickle it. You take it out behind the woodshed, bang it up a little bit increase the vibrational energy of these gas phase cations, and out pops a monomer subunit of this homotetramer, and its charge greatly decreases from the parent ions. It's a threefold increase in charge, so it steals a bunch of charge from the intact complex, setting you up for top-down mass spectrometry of subunits. And we've gone as high as the proteasome in doing this. And so if you do that with enolase, it's a magnesium-containing dimer. Here's the proteoform landscape of enolase from a human cancer cell line grown in a dish. It's basically N-terminal acetyl and not. And then, in the database, you have acetyl phosphomalonyl. We just don't see them to about 5% stoichiometry detection limit. So there's people who believe that primary metabolism, this enolase is part of primary metabolism. There's a couple big science papers that came out about acetylation, and that could be regulating the metabolic flux and things, and that's still true, but to what extent, and how general is it? Okay, so I'm giving other people versions of what we've covered today, and I proposed a project back in 2012, and just 2 weeks ago in Orlando at the HUPO conference, Human Proteome Organization, we socialized this, and my sense is that it's the receptivity of the communities is starting to increase, and the belief that we can actually do a proteome project, cell-based, so whatever comes out of the cell atlas, determine proteoforms to a given depth in each cell type. And we got a sliding start on this thanks to Paul Allen, and here I just have to stop from the news this week. He passed away, and the world lost a great idea, man. It's just plain as that. And I'm going to further just come clean with, that cosmology analogy? It actually started at an Allen Frontiers symposium by seeing people. It was someone else who told me about Tycho Brahe, and so I'm just parroting out what others. Being around great other people stimulates people, and this is an example. So this is a project using denatured top-down 30,000 proteoforms in different cell types of the hematopoietic system. I'm going to just move to close here. So top-down proteomics of specific cell types, circled in green here. We finally got real experts in providing cells of especially rare types. You've really got to know what you're doing. So up in Vancouver, Stem Cell Technologies, about a 1,000-person company, great partners for this, and that's just started. So I really look forward to the future where they can provide cells on a more routine basis. And for these are primary human cells, so we're getting away from cancer cells in the dish. This is the scope of progress thus far. About 15,000 highly curated proteoforms in the Allen Proteoform Atlas. And through our friends at the NSF-funded Magnet Lab in Tallahassee, we're just going to keep on going. And then final slide is what's the value of the information, right? If I'm pitching to you the idea of a genome project for proteins, there better be lots of great spin-outs of this. And so if you're proteoform informed, you can create better assays. And you know, whether you're bottom up or this antibody-based pull downs and then bottom up, if you knew what the landscape was up here, I assert that the value of those assays would improve. Here's where you can circle back to spatially resolved proteomics and cell mapping. So you're familiar with CyTOF, antibody-based technique, but you can do 40 or 50 readouts with amazing sensitivity. That's Scott Tanner in Toronto invented that, and then Gary Nolan has a MIB derivative where he's getting close to atomistic resolution of readout in space using this MIB approach, and he gives a spectacular talk about that. And then just good old clinical ELISAs. You know, the one the horse I always beat up is prostate-specific antigen, PSA. Not a great test. Well, why should it be a great test? We're doing, you know, we're crawling over a big elephant, and we don't even know what it is that we're crawling. You know, you don't know that there's 84 proteoforms that's been mapped of PSA, and some of them could have very different correlations to stage of prostate cancer, stage 1 through 2, 3, or 4. And so just ELISAs, epitope masking, all these things that make ELISAs hard to project into the clinic, I think would improve. And I have more, but I don't have any more time. So I will stop that and just say, borrow from the Genome Project here. You know, this reduction in price that happened, that was when NHGRI really funded explicitly the private sector, and that led to incredible drops in price over this 20-year period, starting at the end of the Genome Project. So it didn't directly come out of the Genome Project, but it was $1 per base, and so I pitch out there that $1 per proteoform would be the analog of that. And you know, with the Human Cell Atlas, let's look at what happened the past 2 years, because I gave Francis Collins came to Chicago with Senator Dick Durbin, and I kind of tugged on his coattails a little bit, and I was like, hey, top-down proteomics? He said, go away, kid, you bother me, but write me. And I wrote him, and he wrote me back, and he said, hey, you know, the Common Fund is a place where we are risk-embracing, and you fit these criteria, and that's the function of the Common Fund. So you all should be watching closely the Common Fund, and he said in his letter reply that you should look at something called the Human Cell Atlas, which was then a Common Fund initiative. It's since become this, and we have program officers here who are behind this, and other Common Fund initiatives like glycobiology, reducing that to practice, and lots of interesting things to study. So track the Common Fund, because they're spinning out RFAs on big, tough problems all the time. And at the end of 2016, you know, Francis said, hey, look at the Cell Atlas. About the same time, slightly before, the Cell Science Institute, run by Rick Horowitz, funded out of Seattle, and the Allen Foundation was spinning up. So there was this kind of focus on cells and cell types. Rick and the Allen Institute want to build movies of really how cells are operating, cardiomyocytes and stem cell biology being a first proof of concept. And now remaining in the NIH Common Fund is this hub map, the Biomolecular Atlas platform, which we could hear about throughout the week. There's an NSF Grand Idea Machine that's for ideas to land around 2026. So you could look into that to improve, to lift up ideas and education mission. And then my last slide, quote from Einstein. Tells you a lot of what's changed. A person who has not made his or her great contribution to science before the age of 30 will never do so. So the older I get, the less I use never and always. And, but this article goes on to show all sorts of examples of people right your age. So all I want to say is, at your career stage, head in the cloud, boots on the ground, and doesn't that just describe young career scientists? So thanks to my group for putting this together and all the hard work we do on Team Purple there in Evanston and Chicago. And I will just need to stop talking now and invite questions. So thanks.
Thank you so much. That was fantastic. I'm wondering, in terms of sort of this dark area, technology is hugely important in terms of what we can see and what we can't see. And a lot of times, I think, as scientists, we look into the biology of a system and we say that we're studying fundamental mechanisms, but many times when you're looking at complex modifications, it's actually, the sort of things that we consider mundane, like separation techniques or ionization or so on and so forth. And have you ever done sort of a more practical indexing like TISNE, which I'm not familiar with, or any sort of PCA analysis to look at the dropout that you see of not, little peptides that you don't see, and what sort of technology is limiting in that sense?
So she wants the gravitational lensing of the dark matter and to get a sense of it. We should, I will admit to being too busy or not smart enough to do that. And it's a great idea. But I think we can characterize bottom-up proteomics studies and put a simple metric on it. If you want to change something, put a metric on it, whether that requires your own lab or a community. So we should put a metric, % total human proteome sequenced. And I'll bet that it's between 5 and 20%, depending on the depth. So that's one way to answer your question.
I just was wondering if you could comment on what modifications do we not yet know about? So are there things that could still be out there?
Yeah, I started my lab with that. It was more than just weigh every protein. I was going to find a novel PTM. And I went to Illinois in part because they are really good at the archaeons, and Carl Woese and Ralph Wolf, and they had found 6 new cofactors. And in fact, there is a new PTM, a pyrrole lysine, the 22nd or 23rd amino acid. That wasn't my work. We have 400 known PTMs in biology. And they're in the RESID database, which is now the UniMod. And John Garavilli is the PTM collector over all these decades. And so there's a lot that's been mapped in rare organisms as well. So there's probably, connectivity differences, cross-linking in weird ways, but I think we're pretty deep into the PTMs discovery.
I know. So I think those 2 gin and tonics last night might have clouded your memory a little bit about what happened. But over those drinks, you mentioned this idea of, was it cooperativity or I forget the term that you coined. It's about collaborating but also competing to get to the
we talked last night about cooperation.
Yeah, that's just your word. And I was wondering if you want to maybe expand on that, because I think it was a really interesting thought. Yeah, exactly. Because I think it relates nicely to your talk and also to the group here that's all at the same kind of stage of their career.
Yeah, so the consortium is exactly that. You've got to get people. There's about 400 members and an advisory board, and maybe with age, some people actually can engage more in cooperation as they get older or younger. Some people can't turn it off, and so you have to deal with all of that and getting together on something big. And so cooperation, yeah, it's an interesting thing to try to wrangle.
I was just interested, so when you have these like situations with these proteins that exist in many, many different isoforms and you have to suss them all out, like what like practically is your approach to even know that you're looking at the right protein? Like do you first like do a purification so you know you have KRAS and then you basically then just run that KRAS sample on the mass spectrometer?
Yes, so targeted top-down or hypothesis-driven work. It took us 18 months to develop this assay and get these KRAS proteoforms and get isoform specific. But it must be like 5% of the resource allocation is geared to do this in mass spec-based proteomics. So maybe when that equilibrates, the pace will pick up and the technologies will as well. Yeah, no, it was just a pull down, just really good antibody. Yep.
Yeah, here. So speaking about what we don't know, when you analyze a proteomic run here.
Yeah. Hi, Jude.
By mass spec, you generally have as a reference the known sequence of the proteome, at least maybe in its simplest iteration. But there is a lot of emerging data that translation sometimes generates proteins that are off the actual coding sequence that we expect for an mRNA. Run translation, frame shifting, nobody really knows to what extent this really happens in a cell except by looking at it with reporters that are very sensitive. So does proteomic illuminate or have people looked at to what an extent the ribosome generates new types of proteins that are frame shifted or that are
So
something
stochastic
different?
biology, or sometimes there's intentional frame shifting. The depth in the proteome required is really needed. Alan Drummond is one of the tops around that could talk to you about that in yeast and what he's observed and extending that to human systems. And proteostasis, what happens when you get old. I mean, you tend to have more bad days the older you get. Maybe that's kind of one takeaway. I don't know. Alan, here, what do you
I guess Judith and I know each other fairly well, so we've talked about some of these things. We tried to answer this question of what sorts of particularly like missense mutations that you would see due to translation errors, because the fidelity of the ribosome is very limited relative to, say, DNA polymerase and RNA polymerase. And I will say, after 18 months in my faculty position, I shut down that project because we were only seeing things that we could possibly interpret as post-translational modifications whose frequencies we did not know versus possible protein modifications. And so ultimately, we have to go very far down the list. And my sense was we weren't quite there yet, that we actually need these sorts of maps first of what's going on biologically to be able to kind of rule in, build some sort of model, because it's not as simple as just getting better at mass spectrometry and getting more sensitive. You actually need to integrate the information that you're seeing so that you can tease apart different hypotheses about what's really going on.
Okay, we'll do one more question here.
Megan,
So I wanted to ask about the function of PTMs, right? We can map them, we have the databases, we can look at their context dependency, tissue specificity. If they're reactive, we can trap them and capture them in all different ways. But it seems like the methods to assign function are not high throughput, right? It's studying individual systems. So I was wondering what your thoughts are of, what high throughput ways could be applied to assign functions. Stoichiometry is a good one.
So I will fit in a hail to P53, discovered by Dan Linzer, 1981, with Linzer and Levine, if you didn't know. Where's my P53 slide? So look at the different views provided by top-down and bottom-up. So here's check one kinase hitting P53. You get 8 phosphorylations in that distribution. You do the same with high sensitivity bottom up, you get like 39 sites, those blue bars, creating a vast potential. But that's that low up, it's the abundance factor. And what I would say to you is an efficient, it's almost too many phosphosites. So to functionalize the PTMs, that as a filter, and look at the dynamics across different states, like with a mutant, and you then look at how the PTMs differ, and that gives you the focal point of which to go assign function, which is an R01 type of endeavor. I don't know how to really, you can go higher throughput, but it's gonna, the technologies to do that are still 5 years off. To get a good profile, good measurement, map the planets, figure out the star in the center of the solar system.